speaker is, and the last but definitely not least, is Fernanda Ponce from INRIA in France, and she'll be talking about wrapping up the IBC project.
Yes, thanks so much for having me. So today I will be talking about the IBC project, or the wrapping up of the IBC project, because we're almost done. Which, if you have not heard of it, is a data set of deep phenotyping. But for this, I need to introduce you to the concept of precision neuroimaging that you have probably heard along these days, because it's been talked about quite a bit. So it's basically that we have a small set of subjects, and we bring them to the scanner many times. So it's basically the extensive sampling of a few set of individuals. Why do we want to do this? Well, because it's been proven that we can preserve subject-specific features, and we can collect how a single brain replies or responds to a very broad set of stimuli. So it allows us to characterize brain responses for single individuals. So back in 2015, the IBC project, the individual birth charting, but we use IBC for short, was born from this idea of probing a lot different complex stimuli in a few brains, so that we can, so that we could create a data set that will serve as a foundational resource, in which we would have a lot of brain responses all together, and we can achieve some deep characterization of individual brains. So what we did we started by doing in 2015, sorry, that's my mouse. Okay, well, anyway. We started to go to different neuroscience labs and ask them to share their protocols with us, so that we could incorporate a different task that other labs were using in our cohort. And this led to the final data set that we have collected, which is a high-resolution fMRI data set that has more than 80 cognitive tasks. So basically, we started with 13 subjects that would come to Neurospin in France many times a year to perform different tasks, all of these tasks that we collected. So, but as you can see, this took a long time. Actually, we finished data collection last October. So it was 8 years of data collection, so that subjects would come very frequently. But life happens, so some subjects dropped out as expected. So at the end, we collected 50 hours of fMRI data for a set of 8 subjects. So what is very interesting for us is that we could track, since we had tasks from all around the world, we can see how different labs track the same concept. For example, here I'm just going to show you data from subject 4. We can see that, for example, in this task by Stagliani and colleagues, it's a functional localizer for the visual system. So here I'm just showing you an example of a body localizer, so showing images of bodies. And we can see some pattern for the specific subject. But it's not the only task that has this kind of concept that we included. The HEP working memory task has also a similar mapping of this concept. And also the task that we got from Van Duffel and colleagues, it also includes this concept of bodies, but across different modalities. For example, audio, tactile, visual, and it's not just human bodies, but monkey bodies. So for us, it's very interesting how can we explore different domains by the way that different people have done it. And also, as you might imagine, very important for us is also the specific subject specificity. So across different subjects, we can see kind of similar patterns of, as you would expect with this kind of task, but we can see how like the responses or the extent of these patterns is highly individualized. And here I'm just showing you more subjects, but it's the same idea. So here I'm just showing you, I have just shown you 3 or 4 contrasts, but as I told you before, we have more than 80 tasks. So this led to more than 700 contrasts. So here I am showing you a similarity matrix of 346 contrasts that we call main contrast. It's just a denomination that we did for those contrasts that we think they are probably more interesting or main, that they have like probably more statistical value. So, so we take all the contrast maps and we calculate the similarity between each other. But it's not the only way that we have to characterize these contrasts. We also annotate them using concepts from the cognitive atlas. And what I mean by this is, for example, for the matrix on the right, we take the statistical maps that are derived from the contrast, and we correlate them with each other for all subjects. And on the left, we compare how similar are the different tags that we gave to each one of the contrasts. So here, for example, you have the bodies and the lipase that we gave it to them, and the other contrast, which is bodies minus all the rest of categories, so we can eliminate a lot of tags, and we just get like the very significant ones. And we also calculate the correlation or the similarity of these two representations. But here I'm showing you 346 contrast maps, so it's a lot of information. But what happens if we try to separate or see how, if we can form some clusters of tasks? So we run an embedding so that see how these contrasts distribute in the space. And what I'm showing you here is exactly what I showed you before, but it's just one matrix. So the lower triangle is the similarity of contrast maps, and in the upper triangle is the similarity of concepts. So one of the clusters that highly comes out are these tasks that you can already see that it has a strong auditory component. The first two are auditory localizers, and the other two have very much strong auditory stimulation. So we can see how already the similarity of these representations goes a bit up. Another of the clusters that really goes up is these. We can see that it's a lot of motor, very motor responses. And we have the same for different domains. I'm just showing you these other two clusters that come up. One has a very strong emotional component, and the other one is a bit divided between language tasks and working memory tasks. So we calculated the correlation of the representation of the intra and inter-subject correlation of all these maps. So we can see that, as expected, the intra-subject correlation is higher, but the distance between them is not that big. So that kind of suggests that the inter-subject variability is moderate. As I mentioned at the beginning, these kind of approaches allow us to get some individual topographies or very individualized representations of the brain. So using the approach by Abraham and colleagues, we fit a dictionary model to to contrast maps to derive some components. So if we fit, for example, this task, we get some very individualized topographies in which each one of the components is labeled according to the contrast with the highest loading. And what I'm showing you in the bottom left is the group mean. And finally, one of my favorite things that came up with this project is the brain cockpit that is hosted on the eBrains platform, in which you can click on any location on the brain, and it will tell you the activation or the C-score that that particular location had across all maps. So it's very interesting to see, you click on one location, and it's very interesting to see how the CMAP moves, and you can filter through subjects, you can plot the group mean, you can plot different surface meshes, and you can change or filter the contrast, just the task, etc. And you can have a lot of visualization. So it's quite a nice way to visualize the signature of all of these locations. Something very important for us is that the data was always meant to be open and accessible. So the data, raw data, pre-processed data, and contrast maps, it's all available. Our main platform right now is eBrains, because some years ago we started with OpenNeuro, but as we already heard, the regulations kind of changed recently. So we still have some data in OpenNeuro, and we still maintain it, but we have migrated mainly to eBrains, in which the data is available. We only require people to have an account. It's a bit of like a long curation process, so it's not all the data available yet, but we're working on it. We have just like some curation requests. We also, all the protocols that we collected from all around the world, we have them available. We have some demos that you can see just to see how the tasks look like. And we also have published the routines and pipelines that we use to analyze the data. And finally, we have a very extensive documentation because there are a lot of stuff that I couldn't cover here, like a lot of details on the subjects, details on all the tasks. We describe all the tasks, what we chose as contrast, etc. So it's in the documentation, and we also publish some articles with this information. So the last paper that came out describes all the naturalistic stimuli that we included, some movies and some retinotopic tasks. So in short, the IBC project, it's a data set that has 50 hours of fMRI data, which derived in more than 700 contrasts, all of them that were labeled using more than 100 cognitive concepts. We also acquired some anatomical and diffusion data. And important for us, we also included some movie watching and video gaming in the scanner. That was quite fun. As you might imagine, this project has been going on for a long time, so there's a lot of collaborators and a lot of people who have been involved. We want to thank all the labs. Probably, if you've been working on this for a long time, we'll probably reach out to you to get your protocol, so thanks very much. Big thanks to the participants, because I don't know if you imagine coming to the scanner through all these years, through COVID and through everything. So it's a huge effort on their side. And thanks to our funding, and with that, I thank you.
So one last question before we wrap up, and we'll be able to finish on time. Any questions from the floor? We're keeping an eye on the app. There's none on the app. No? No questions? Well, thank you so much to Fernanda, and let's thank all the speakers again for their incredible efforts in making science more open. Thank you. There were lots of questions that we couldn't answer because of time, but please come to meet the speakers and ask them later in the coffee break. Thanks again.